So let's now we go into the details of different reactions of gluconeogenesis. I want your real focus here because these are complicated. I mean, these are not easy to understand reactions. सबसे पहले you can think gluconeogenesis, because it's the only formation of glucose, then it can be simple reversal of the glycolysis process, because glycolysis is the breakdown of the glucose into pyruvate. So why can't we simply reverse the glycolysis process and move in the reverse direction from the pyruvate towards glucose formation? Yes, it might be a very easy way, but it's not possible because of many problems, because of many limitations the living systems have. So gluconeogenesis in this way is a process which circumvent these limitations. So first of all, we see the, if you can see, this is the whole is the glycolysis process, and there are certainly 7 reversible reactions. आप देख सकते हैं कि there are reversible reactions which can easily be, I mean, used by gluconeogenesis to just reverse the direction of the for the formation of glucose. Fine, but not all the reactions are reversible. There are 3 irreversible reactions also. मिसाल के तौर पे पायरोवेट से फास्फोनिल पायरोवेट का बनना, इट्स रिवर्सिबल. फ्रुक्टोज 1,6 बाइफॉस्फेट से फ्रुक्टोज 6 फास्फेट का बनना, आई मीन फ्रुक्टोज 6 फास्फेट से फ्रुक्टोज 1,6 बाइफॉस्फेट का बनना, ये इरिवर्सिबल. ग्लूकोज से ग्लूकोज 6 फास्फेट का बनना इरिवर्सिबल. और मुझे यहाँ थोड़ी सी लेट मी करेक्ट इट, फास्फोनिल पायरोवेट से पायरोवेट का बनना ये इरिवर्सिबल है. Okay, these are irreversible. You cannot simply reverse them because they are irreversible. What does it mean irreversible? That thermodynamically the reversal is not feasible. तो फिर हम क्या करें? We have to have a machinery which can reverse them. Certain enzymes, that machinery I mean. So what? First of all, so one of the first reaction would be that we can somehow reverse the reaction from pyruvate towards the phosphonyl pyruvate, but there is no enzyme which can directly convert pyruvate into phosphonyl pyruvate, so we cannot this cannot be reversed. So because this cannot be reversed, then one way is that pyruvate is first converted into oxaloacetate, and then oxaloacetate can be converted back to the phosphonyl pyruvate, and then it will become part of the reversal of the glycolysis process. But the thing is that the oxaloacetate, though it can be formed, but we don't have any shuttle service in the cytosol which can really which can really involve in the transportation of the oxaloacetate. So oxaloacetate have to be in the mitochondria. So what will happen? For example, let us see here that in the mitochondria, now keep in mind there are 2 different junctures here, 2 different environments. We are talking about cytoplasm and we are talking about mitochondrial environment. The pyruvate firstly is produced in the cytoplasm because of the glycolysis process. Then that pyruvate, of course, it has certain transporters which let it to enter into the mitochondria, and that pyruvate is converted into oxaloacetate. And we know that, and the enzyme which is involved is pyruvate carboxylase. And we know that oxaloacetate, through the activity of phosphorylated phosphoenol pyruvate carboxylase can be converted back into phosphoenol pyruvate. But the thing is that we don't have any machinery in the mitochondria which can take phosphoenol pyruvate outside of the mitochondria into the cytoplasm where the gluconeogenesis process. So first we have to convert the oxaloacetate into some moiety which can be transported out of the mitochondria into the cytoplasm. That is why the oxaloacetate is converted first into malate. Now once the malate is formed, the malate can be transported outside of the mitochondria because we have the system, the living system have the proteins or shuttling proteins for the transportation of malate. Once the malate comes out, the malate is then converted into oxaloacetate. Now we have oxaloacetate available in the cytoplasm. Fine. Now this oxaloacetate is acted upon by the phosphoenol pyruvate carboxykinase, which convert it into phosphoenol pyruvate. Once the phosphoenol pyruvate is formed, then it can become the part of the gluconeogenesis process. Then it will be simply involved in those reactions which only need to be the reversal of the glycolysis process. For instance, so this is phosphoenol pyruvate formed from up till here till the glycerol 3 phosphate formation. There will be a simple reversal of the process of glycolysis. Also, you should keep in mind that this is a very, very energy demanding process. I mean, there are lots of conversions you have to brought about before you get the phosphoenol pyruvate. I mean, so many circumventions, so much need of the energy. But eventually, and it tells you also the importance of glucose. कि आपका लिविंग सिस्टम इतनी एनर्जी इस्तेमाल करने में उसमें कोई मसला नहीं अगर उसने ग्लूकोज बनानी है. ये बताता है कि ज़्यादा एनर्जी चाहिए हो भी तो ग्लूकोज बननी चाहिए. इट्स सो इम्पोर्टेंट. यहाँ कुछ क्वेश्चंस हैं. ऑब्वियसली ये एक थोड़ा सा आपके दिमाग को ज़्यादा मांगता था ये वाला टॉपिक कि किस तरह हम सर्कमवेंशंस करते हैं डिफरेंट पाथवेज की ताकि हम ग्लूकोनेोजेनेसिस को चला सकें. तो ऑब्वियसली आपके पास क्वेश्चन भी फिर उतने जानदार होने चाहिए. Why pyruvate is not converted directly into oxaloacetate in cytosol? साइटोसोल मतलब पायरोवेट को क्यों माइटोकॉन्ड्रिया में जाने की ज़रूरत हुई और ऑक्सैलोएसीटेट को उसने बनाया, उधर जाके वो फिर मेलेट में चेंज हुआ और फिर बाहर आया और फिर ऑक्सैलोएसीटेट में चेंज. क्यों नाम सीधा सीधा साइटोप्लाज्म में पायरोवेट वुड हैव बीन कन्वर्टेड इनटू ऑक्सैलोएसीटेट. व्हाट वाज द प्रॉब्लम देयर? Why the mitochondrial oxaloacetate not converted into pyruvate phosphoenol pyruvate PEP डायरेक्टली वो उसमें चेंज हो जाता? उसमें क्या मसला था? क्या प्रॉब्लम हो सकती थी उसमें? अभी उधर से चेंज होता, PEP बाहर आता और साइटोप्लाज्म का हिस्सा बन जाता और ग्लूकोनेोजेनेसिस का पार्ट बन जाता. What is the importance of reversibility and irreversibility to understand the process of gluconeogenesis? What do you think? Is it gluconeogenesis is more energy yielding or more energy demanding? And finally, इन्हीं में एक क्वेश्चन एक और एडिशन कर लें, लाजमी नहीं, जैसा कि मैंने कहा था पहले भी, इट डजंट हैव टू बी ओनली दिस लिस्ट ऑफ क्वेश्चन. एक और भी कर ले. आप अपनी तरफ से क्या डिज़ाइन करेंगे सिस्टम को, किस तरह के इससे ज़्यादा आसान तरीके से आप क्या समझते हैं कि नेचर क्या करती, व्हाट नेचर शुड हैव डन दैट द ग्लूकोनियोजेनेसिस प्रोसेस was much more easier. At least this formation of phosphoenol pyruvate from pyruvate would have been more easy. What nature could have done for that? आप क्या नेचर को सजेस्ट करेंगे? आप क्या सजेस्ट करेंगे नेचर को क्या करना चाहिए?